Good afternoon, everyone, and thank you for joining this National Bleeding Disorders Foundation Wednesday webinar. My name is Fiona Robinson, and I'm the series host. In today's webinar, we will learn about advancing hemophilia A treatment, latest insights on factor VIII expression, durability, and gene therapy. This webinar is being recorded and will be available to the community on the NBDF website, bleeding.org, shortly after the webinar. We encourage you to ask any questions you may have today. You can ask your question by using the Q&A feature at the bottom of your screen at any time. We will monitor these questions, and following our speaker's presentation, we will put your questions to them. Today, we are joined by Dr. Lindsey George, Assistant Professor of Pediatrics at the Perelman School of Medicine at the University of Pennsylvania and Director of Clinical In Vivo Gene Therapy at the Children's Hospital of Philadelphia. I'd like to thank Dr. George for joining us today, and I will turn things over to her now to get us started.
All right, excellent. Thank you, Dr. Robinson. I appreciate it. And thank you all for joining, particularly right for the holidays. I know it's a busy time of year. So it's really an honor to be with you all today. I'm going to focus, even within the context of hemophilia gene therapy, to talk about hemophilia A gene therapy and specifically addressing some of the considerations around durability of factor VIII expression and what that may mean for potential next generation approaches to hemophilia A gene therapy. And so what I'll focus the talk about are essentially four main components. One is just speaking about long-term factor VIII expression after use of adeno-associated viral vectors or AAV vectors for gene transfer. Potential future approaches using a factor VIII variant that has improved function for next generation approaches. And then think about the importance of factor VIII expression durability as it relates to development of antibodies to AAV vectors and some approaches to potentially get rid of these antibodies in the future. And then just I'll conclude with some broad-based considerations that I think is all important for all of us to consider about when we sort of build on the successes of where we are in gene therapy to make these approaches even better. So I don't have to tell all of you what hemophilia is. I'll just remind, again, I think many of you on this webinar know certainly much better than me what it means to have hemophilia and what the symptoms are. I'll just note briefly when thinking about hemophilia that the current recommended standard of care is really to convert the severe phenotype to that of a mild phenotype with the WFH guidelines targeting factor troughs of less than or equal to 5%. And then, of course, there are a number of therapies outside of factor replacement that are currently in use, most notably emicizumab, which in many ways is the de facto standard of care for hemophilia A, as well as multiple approaches in various phases of clinical development or recently receiving FDA or EMA approval. Focusing on hemophilia gene therapy efforts, there are a number of approaches that have been tried in the clinic. I'm going to focus, as I noted, on the use of adeno-associated viral vectors or AAV vectors. AAV vectors for hemophilia are systemically infused, targeting the liver to express either factor VIII or factor IX. The use of AAV vectors, so the idea is here you're targeting cells in the liver, specifically hepatocytes for expression of factor VIII or factor IX. This is where factor IX is made normally or endogenously, but is not the endogenous site of factor VIII synthesis. Transduction or the entry of AAV vectors into entry of AAV vectors into the target cell of origin, as well as subsequent encoding and delivering of a genetic payload to the nucleus for expression of factor VIII or factor IX, as you might expect, is a very complex process with a lot of opportunity for biological variability, which is likely contributing at a minimum in part to some of the variability we see in clinical trials. So I'll focus mainly on use of adeno-associated viral vectors for hemophilia gene therapy, but briefly before we venture on that journey, I do want to note that there are a number of efforts using lentiviral vectors to target hematopoietic stem cells ex vivo or outside the body. So the idea here is you have hematopoietic stem cells, you use a lentiviral vector. And most notably, these efforts are for hemophilia A to deliver a factor VIII gene to be expressed in hematopoietic stem cells. And then these cells are delivered back to patients. This does require conditioning. The regimens are akin to what is used for bone marrow transplant with a bit less myeloablation. And so that I think presents a complication a bit for the hemophilia population as it relates to potential for safety. But I will say, importantly, there are at least 2 of these trials have had clinical data presented and looks quite promising. Most notably, this trial outlined here was recently published in the New England Journal, and you can see the systemic factor VIII activity observed in the first 5 participants to receive this therapy. And so we'll look forward to hearing more about this. But as I noted, we'll focus over the next approximate hour mostly on AAV efforts. So this is a table of ongoing AAV gene therapy work. Those highlighted in red or bold in red here are licensed or FDA approved hemophilia B vectors. Of course, there are 2, Beckvez and Hemgenix. I'll use the trade names if that's all right. For these 2 vectors, and then in hemophilia A, there's a single licensed vector, the Roctavian manufacturer by BioMarin, and then there are a number of additional approaches that are ongoing, are in various phases of clinical development. One, I think, for the rest of the talk, we'll really focus primarily on hemophilia A efforts. I think one of the major outstanding questions that's come out of clinical trial is this question around durability of expression. So these are 2 figures of aggregate clinical trial data of sustained factor expression for either hemophilia B efforts or hemophilia A efforts. And most strikingly, you can see that the trials that have had sustained factor IX expression outside that initial couple-month window really have had, quite durable expression across all of the trials. You can see multiple trials have demonstrated stable expression multiple years out. And that's not necessarily what we're seeing in hemophilia A gene therapy. Most notably, the 2 trials that have achieved initially normal or near normal levels of factor VIII expression have observed this year-over-year decline in factor VIII expression of some unclear etiology at this point in time. And then there's at least a single vector that with published data that have demonstrated more modest initial levels of expression, but these levels of expression are stable over time. I think actually what's really important though is when you delve into the clinical trial data a bit further, this concept that more modest levels of factor VIII expression can indeed be durable is demonstrated across at least 4 trials. So this is the data from the Spark trial that I had just shown you, looking at factor VIII levels at year 1 versus year 2. This is just looking at the mean of the difference between the factor levels from year 1 to year 2, and you can see the mean of the difference is right around 0. So there's really not much of a change. And then these are data that have been presented only in abstract form of additional vectors, but you can see here, again, looking at factor VIII activity over time on these 2 trials, and you can see levels of factor VIII expression in the range of moderate or mild hemophilia A have been demonstrated to be durable across a couple trials. This is the figure I had shown you previously, but this table here just summarizes the trials that have shown durable factor VIII expression exclusively at this point in the range of moderate to mild hemophilia A. That includes published data from Spark, which I just showed you. The lower dose cohorts of the Pfizer Sangamo developed vector did have what looked to be durable factor VIII expression at the lower dose cohorts. That, of course, those doses were not taken forward into phase 3 clinical trial. And so it would be really important to understand the long-term follow-up of the phase 3 clinical trial participants to see if they have stable expression. And then data I just showed you that have been presented in abstract form only. Consistent actually with these observations in clinical trial that levels of factor VIII expression in the range of moderate or mild hemophilia A can be durable. Before success in humans, there was success in canine models of hemophilia A, and so there have been dogs that have been followed for up to a decade who have demonstrated single-digit factor VIII expression that did not decline for up to 10 years. So these together generated the hypothesis that durability of factor VIII expression may actually be dependent on the amount of factor VIII expressed. And so we were interested to see if we could model clinical trial observations in mice, and in doing so, then we could try to understand the mechanism of factor VIII durability of expression, and then hope to improve upon future generation efforts, building on current successes. So what we did is we took hemophilia A mice, we treated them with a AAV8 vector to express factor VIII. The vector was made in mammalian cell culture, so that's HEC293 manufacturer. This is mammalian cultured manufactured vector. We treated animals to express factor VIII in the range of mild hemophilia A, normal levels, and elevated levels of expression, and we followed them out to 72 weeks. When we looked at this is looking at their plasma factor VIII levels over time out to a year and a half. You can see here that initial expression in the range of mild hemophilia had stable levels of expression. Again, initial normal levels of expression were stable, but if you had higher levels of initial expression, the mice lost about half of their expression on a year and a half of follow-up. And this is a summary of the same essential plasma antigen values. This is week 8 steady state factor VIII expression levels versus week 72 values. And again, initial elevated levels of expression significantly declined on a year and a half of follow-up. And we took those same animals and we had harvested their livers, something, of course, you could do in mice, but not in people. So again, we're interested in sort of, can we delve into the mechanistic basis of clinical trial observations? And we looked at their livers and quantified factor VIII mRNA as well as the vector copy number. So the concept here is that this could give us some information about the efficiency of transcription of the donor DNA template. And the concept would be if you have vector copy numbers there, so you have evidence of vector DNA there, but not factor VIII mRNA or factor VIII expression, that this may be consistent with gene silencing. But this is actually not what we saw. So this is looking at the ratio of factor VIII mRNA to vector copy number across the 3 different expression cohorts, and that ratio was the same. And notably, this elevated cohort at week 72 had this significant decline in expression. So the data demonstrated that in mice, the durability of factor VIII expression correlated with the amount of factor VIII being expressed, and there was not evidence of gene silencing. A talented research associate in my lab wanted to make sure that this was not related to the actual vector expression cassette, so actually repeated this experiment with another vector, again, manufactured in mammalian culture, and essentially saw the same results, that initially high levels of expression were not stable while lower levels were. And again, we didn't see evidence of gene silencing, and so the data were consistent across 2 different vector cassettes. So acknowledging that we didn't see evidence of gene silencing, we next wanted to look to see what the vector copy numbers were doing over time. You know, the hypothesis being that perhaps the loss of factor VIII expression is related to losing factor VIII levels over time. Vector copy numbers over time. So we had livers from animals treated treated and followed for 72 weeks, and we wanted to look at an earlier time point. So we went back and treated animals with the same vector cassette, expressed factor VIII at the same levels, either range of mild, normal, or elevated factor VIII. And we wanted to first just make sure the animals that had livers harvested at week 8 versus week 72 were antigen matched for the same levels of expression, and they were. And then when we looked at the vector copy number at week 8, we saw, as expected, this is looking at vector copy number by the level of factor VIII expression, as you may expect. If you had more factor VIII expression, you had significantly greater vector copy numbers over time at the earlier time point at week 8. But when we looked at week 72, again, where we'd seen this decline in expression in the initially elevated cohort, what we saw is that there was no difference in the vector copy number values observed for the normal versus the elevated expression cohort. And this was consistent with what we saw with their levels of factor VIII expression, which did not differ. And so these data suggested that the loss of factor VIII expression was due to loss of vector copy number. So then the question is, why were these animals losing copies of the vector? And, you know, this has been a mammalian expression of the factor VIII protein is inefficient, in part due to factor VIII protein misfolding and aggregation in the endoplasmic reticulum, where factor VIII is initially made. And this triggers something called an unfolded protein response that unmitigated can trigger pro-apoptotic pathways or cellular death pathways. And so we consider the possibility that if you had increased levels of factor VIII expression, you may be inducing evidence of ER stress and unfolded protein response that potentially could be loss of result in death of the transduced cell. So we took at those same livers and looked at mRNA values of proteins that are upregulated in the setting of an unfolded protein response. Those are CHOP and BIP. These are the CHOP values. What we saw is that CHOP values at the earlier time point were significantly elevated in the mice that were expressing elevated levels of factor VIII, but not lower levels of expression. And then at the later time point, both the normal as well as elevated factor VIII mice expressing the normal or elevated levels of factor VIII both had significantly elevated CHOP values. I by no means think this is definitive to suggest that this is what's going on, but I think it at least warrants further investigation. And I think one of the complications is we don't know the exact time frame to be looking for this response. We next looked at the idea here is that loss of vector copy number is due to loss of those transduced hepatocytes. We had plasma samples, so we looked at ALT, which is done, mouse ALT the assay is done very similar to what you do for patients. So we looked at ALT values over time in our expression cohorts, and what we saw is that initially mice that had elevated factor VIII levels as well as the normal expression had significant elevated ALT values relative to more modest, mild levels of factor VIII expression at the later time points. And interestingly, we saw those significantly elevated ALT values at the same time that we temporarily had saw this decline in factor VIII expression. So again, I think there are multiple limitations to the interpretation of these data. As I noted, including unclear potential kinetics of this unfolded protein response, and the data thus far are inconclusive, but they may support this hypothesis that this factor VIII expression level dependent durability may be the mechanism may be due in part to a potential triggering of an unfolded protein response, which results in loss of transduced cells. So we're interested in probing into this deeper, which I think is going to be difficult, but we hope to gather some additional information. I just want to note some work by other groups I think that's also important. This is data actually published by the research group at BioMarin, which I think is really important. So they had looked at expression, used an AAV vector to express a different protein. This is human alpha-1 antitrypsin. This is naturally endogenously made in hepatocytes. So they used an AAV vector to express this protein. They expressed it at higher than normal levels. And what they saw is the vector is made either in the baculovirus system, this is an insect cell line, versus mammalian cell culture. And both they saw a decline in expression of the transgene, again, alpha-1 antitrypsin by about half on the approximate 1 year follow-up window. What they similarly saw, this is data from the animals that were treated with mammalian-produced vector. What they saw over time, this is looking at vector copy number values over time. They saw a significant decline in vector copy numbers over time. And then they saw a direct correlation between expression and vector copy numbers, as you might expect. But what's important is, like we had seen in, so this is consistent with what we observed with factor VIII expression in mice, where animals appear to lose vector copy numbers over time. And this was true for mammalian-produced vector as well as the baculovirus-produced vector. However, one thing I think is important here, this is the baculovirus-produced vector. If you look at the relative amount of protein expressed by copies of the vector, you would expect a positive correlation, which is what you see here in the mammalian-produced vector. But what you see here is that it's not, obviously not all of the values were positively correlated, suggesting that the amount of protein expressed here is less than what you might expect with the amount of vector that's present. And when they looked at markers of genomic accessibility, they were significantly reduced at the later time points for the baculovirus-produced vector. And so what these data suggest is that mammalian-produced vector, consistent with our observations, the loss of the transgene expression correlated with loss of vector copy number in mice. However, manufacturing using the insect cell line manufacturing, loss of expression correlated with loss of vector copy number, as we had observed. But they also saw epigenetic modifications with reduced genomic accessibility, suggesting some element of gene silencing may also be responsible for loss of expression. So all the data I've shown you so far are in mice. So what about in people? These are clinical data presented from the group from BioMarin. This is, just to be transparent, this is data I was not involved in. This is taken from a publicly available presentation and were presented at a conference in Europe earlier this year. But the group had taken biopsy samples of 12 participants in their clinical trial that received the vector dose that's now the licensed vector dose for Roctavian. The liver biopsy samples were taken 1 to 4 years post-vector. And from those liver biopsy samples, they quantified the amount of vector as well as the amount of factor VIII mRNA. So that's factor VIII transcripts. Generally speaking, in 10 of the 12 participants, there was as you may expect, a positive correlation between how much vector was there and how much factor VIII mRNA was there. So positive correlation between, more vector, you get more mRNA, more transgene expression. However, 2 of the participants had low levels of vector copy numbers, but didn't have detectable factor VIII mRNA. Importantly, concurrently, as you might expect, if there's no factor VIII mRNA, there was no longer detectable factor VIII expression at the time of the liver biopsy. So these are looking at the factor VIII activity over time of those 2 participants. And what I think is important here is that the 2, these men initially had a factor VIII expression, but lost it within about a year post-vector. And by the time the liver biopsies were done, indicated in these red arrows, they no longer had detectable expression, suggesting that their initial loss of expression was due to gene silencing. What I think, though, is important, let's see if I can advance, there we go, is that the timing that these men lost expression is actually within that first year post-vector. And so I don't know how to have this necessarily be an observation for what's been observed among the whole cohort of this year-over-year decline of expression. So in general, I think these data are actually really quite important. That again, 2 out of the 12 participants lost all expression with less than a year post-vector administration. Those patients had the lowest vector copy number in factor VIII mRNA. But these data thus far don't explain this year-over-year decline in factor VIII expression, at least to the best of my understanding for the data that are available so far. So to summarize what we've discussed so far around factor VIII durability of expression, I think it's important to acknowledge that clinical trial data as well as animal data support that durable factor VIII expression may be dependent on the actual amount of factor VIII protein you're expressing. Thus far, we've observed that factor VIII expression in the range of moderate or mild hemophilia A can be durable in mouse models, canine models, and of course in people. I think for further understanding this, I think the data support that the mouse model may be useful to sort of tease out and mechanistically understand this better. Thus far, the data support, at least with mammalian-produced vector, loss of initially higher levels of factor VIII expression may be due to loss of vector copy number. And then there are data support with a baculovirus manufactured vector that in addition to loss of vector copy number, the gene silencing may also contribute to loss of factor VIII expression. So where does this leave us in a clinical context? I've focused primarily on durability, and for limitations on time, I can't necessarily talk about all of these points. But I think as all of this sort of points that we'd like to improve upon in clinical trial, but thus far, available data has generated this hypothesis that perhaps we might be able to improve upon the long-term efficacy of hemophilia A gene therapy using AAV vectors by incorporating enhanced function factor VIII variants. So the idea would be to express low levels of protein that multiple trials have demonstrated can be durable, but have the protein work better. So the idea here is you have low levels of antigens to permit durability, but because the protein works better, you may be able to approximately normalize factor VIII function. And this also may permit the use of lower AAV vector doses to minimize AAV-related toxicities, namely immunological responses that have required immune modulation. I'll just briefly note, of course, this approach of using enhanced function variant has been universally incorporated into clinical efforts currently in hemophilia B gene therapy. So all hemophilia B clinical trials, they're actively either enrolling or licensed. Again, there's 2 licensed hemophilia B vectors. They incorporate the factor IX POTO transgene that has about an 8-fold improvement in specific activity. And this is just looking at the aggregate clinical trials, looking at factor IX activity using either the wild type protein, and then of course, if you incorporate this factor IX POTO protein, you have a marked improvement in achieved factor IX activity. So can we do a similar approach for factor VIII, which would have the added advantage potentially or we hypothesize of permitting durability of expression? There's no such occurring enhanced function factor VIII variant, and so we and others have pursued options to rationally bioengineer the factor VIII protein for improved function. This is a summary table of various efforts to do this. We're particularly interested in how the factor VIII protein is inactivated. There are 2 mechanisms, either dissociation of one of the domains of the factor VIII protein, A2 domain dissociation. This inactivates factor VIII. The other way is that the protein 8A is cleaved by activated protein C, and this also inactivates factor VIII. So we wanted to probe these two pathways to see how this may improve factor VIII function. This is the domain organization of the factor VIII protein. We introduced mutations that either disrupt APC cleavage, which is outlined here in these red. These are arginine to glutamine mutations at the two described APC cleavage sites, or stabilize the A2 domain. So these are mutations originally described by FVIII's group some time ago. These improve electrostatic interactions with the A2 domain so that it's more stable. So we made this cadre of proteins. So here, and I'll call this factor VIII-QQ. This protein is resistant to APC cleavage. This protein, factor VIII-VV, just cannot, the A2 domain is stabilized. So you eliminate that, or you reduce that mechanism of VIII inactivation. Or this protein is the combined mutation. So you inhibit APC cleavage and you stabilize the A2 domain. I'll skip this for the sake of time, but we did biochemically characterize the proteins. But I just wanted to show how these, sort of as a direct comparison between all of these proteins, how they worked in a mouse model. So we took hemophilia A mice, we infused these proteins at increasing doses, and then we quantified blood loss. So less blood loss means the protein's working better. So this is our wild type. This is our initial comparator. This is the protein that has a stabilized A2 domain. You can see it works better, about 2.5-fold better than the wild type protein, and that's consistent with what others have also observed. If you eliminated APC cleavage, it works somewhere on the order about 4 to 5-fold better. And then if you combined eliminating APC cleavage and stabilizing the A2 domain, the protein worked markedly better, about 12-fold better than the wild type protein, at least in this assay. And so I think this clearly supports that if you disrupt inactivation of factor VIII-A, this is going to improve function of the protein. We've done the most work with this variant. So the idea here is you eliminate APC cleavage, but you still maintain that the other mechanism of inactivation, the A2 dissociation. The obvious concerns with using this is that this may confer prothrombotic risk. So I don't think there's free lunch in anything with hemostasis. So we wanted to do some preliminary safety studies to think about prothrombotic risk. So we took hemophilia A mice. We expressed factor VIII in the range, again, of mild, normal, or elevated levels of expression. This is the wild type protein, and this is the QQ protein. These are just levels of expression. And I'll just summarize a lot of data. We just looked at survival of these animals, and with the idea that we wanted to ensure that the animals expressing the factor VIII QQ protein didn't have decreased survival relative to animals expressing the wild type protein, and we did not see that. For the sake of time, I'm going to just, actually, yeah, I think this is an important point. So mice do not, they're pretty resistant to thrombosis. And so we wanted to push the system a bit further. And so we took hemophilia A mice and we back crossed them on the background of factor V Leiden. So factor V is a protein that's homologous to factor VIII. It's also a cofactor in the coagulation cascade. Factor V Leiden is disruption. The factor V protein is also inactivated by APC cleavage, and factor V Leiden has disruption of one of those APC cleavage sites. So the concept is that if we took these hemophilia A mice on the factor V Leiden background, so their factor V is resistant to APC cleavage, and we wanted to see what would happen if we also made their factor VIII resistant to APC cleavage. We know if mice don't have APC anticoagulant function, it's fatal. So we honestly expected that if we overexpress the factor VIII QQ protein on the factor V Leiden mice, we basically make the animals functionally APC deficient. And as expected, what we observed, this is looking at survival over time. If you express the factor VIII QQ protein in the range of normal elevated levels of expression, the animals did not survive, whereas the wild type animals did. But importantly, if we express the factor VIII QQ protein at lower levels in the range of mild hemophilia A, there was no difference in survival relative to the wild type mice, which is actually the range that you would target in the clinic. And so actually, I think this was a really important experiment for showing that APC regulation of VIII-A is clearly physiologically relevant. But importantly, translationally, if you just express lower levels of factor VIII QQ protein, the animals didn't have any survival disadvantage, and that would certainly be the goal if you were to translate this into the clinic. The next point is that this factor VIII QQ protein has 2 amino acid changes, so this raised the theoretical concern that it could be pro-immunogenic and raising the risk of potential inhibitor development. So we wanted to see about the immunological response to the factor VIII QQ protein relative to the wild type protein. We used a mouse model originally developed at the Children's Hospital of Philadelphia about 20 years ago. These mice have immunological tolerance to the human factor VIII protein, and we were interested in seeing if these mice already had tolerance to the human factor VIII protein, and if you express the factor VIII QQ protein, did it break this tolerance? And that would be akin to what we would do in clinical trial as an initial first step, meaning take people that did not have inhibitors and ensure, from a safety concern, we want to make sure expression of the factor VIII QQ protein did not result in inhibitor formation. And so we did these studies two ways in these mice. So we either exposed the mice to recombinant protein, either factor VIII QQ versus the wild type protein. And this is quantifying a Bethesda titer, so inhibitor titer, on that exposure. And there was no difference in inhibitor development between animals expressing either the wild type protein, animals exposed to the factor VIII wild type versus our factor VIII QQ protein. And then similarly, we took these animals, initially exposed them to factor VIII protein, and then treated them with AAV vector to express either the wild type protein or our factor VIII QQ protein. And again, we didn't see any evidence of, we didn't see any difference in the development of inhibitors. So this suggested, at least in a mouse model, there was not evidence that this might be expression of factor VIII QQ might have increased risk of inhibitor development over the factor VIII wild type protein. All right. I might skip this one portion just for the sake of time, because I wanted to just lastly talk about the context of factor VIII durability as it relates to AAV neutralizing antibody formation. So this is the graph I had just shown you. And I guess the fundamental question is that, quite honestly, having factor VIII protection for, multiple years, I would imagine would be quite promising. However, the concern is what happens when you no longer have therapeutic efficacy. And I think this is a really important point, because what we do know is that once you receive a systemic AAV vector, patients universally develop high titer AAV neutralizing antibodies. So you can think of infusion of an AAV vector as a very effective vaccine against AAV. And these are data from the first men that ever received an AAV vector. They happen to be patients with hemophilia B. There's just 3 patients, but they were followed anywhere from, you can see, 12 to 15 years. We quantified their AAV neutralizing antibody to the infused AAV serotype as well as others. And I think what's really important is that the titers didn't wane over these up to 15-year window. And subsequent efforts have seen analogous findings, albeit with a lesser degree of follow-up. And so I think what this really effectively means is that this may mean that, and these antibodies, I'll just say the other point is that the antibody titers that develop after a systemic AAV vector are a bit of a different kettle of fish, so to speak, than the titers that typically develop after environmental exposure, and that they're typically orders of magnitude higher, and really at higher levels than we've ever seen the ability to overcome with increasing AAV vector doses, etc. And so I think what this means is, at the current state of clinical development, we don't really have a way to have a repeat AAV vector dose. So you really get one lifetime chance of receiving an AAV vector. Of course, this is a problem both with pre-existing antibodies from environmental exposure as well as antibodies, AAV neutralizing antibodies that may develop after receiving an AAV vector. And a number of groups have tried to eliminate AAV neutralizing antibodies. And this is just summarizing approaches that have been tried targeting B cells. So these are what's type of B cell called plasma cells secrete antibodies, which would be the neutralizing antibodies. But the idea is to sort of target therapeutically anywhere along the line of from the naive B cell to ultimately plasma cells that make these antibodies. There are a number of different approaches targeting different B cell compartments as well as plasma cells. And then others looked initially quite promising at depleting actually the neutralizing antibodies or themselves, which are IgG antibodies. So there have been a number of studies in, I suppose, small and large animal models, as well as a couple even that have made their way into people, different approaches that are summarized here. But I think suffice to say, there's no confirmed clinical strategy to reliably get rid of high titer AAV neutralizing antibodies. And therefore, these are anticipated to preclude potentially initial efficacy with an AAV vector, as well as certainly to preclude repeat administration. So we were interested and thinking through the different potential B cell compartments that may need to be targeted to get rid of AV neutralizing antibodies. The concept was to use two different approaches, which I'll outline here. But I just wanted to say what the two different approaches are either targeting these markers, CD20 or CD19. These are markers on B cells, and you can see the different types of B cells that this would be targeting. CD20 through sort of immature B cells through memory B cells, and then CD19 immature B cells through plasma cells. Remember, those are the ones that secrete antibodies, but would not get long-lived plasma cells, which one may hypothesize are involved in AAV neutralizing antibody persistence. The first approach that we wanted to use to target these markers on B cells was the use of CAR T-based therapy. So these therapies are being used for a variety of B cell malignancies as well as many other therapeutics, but there's multiple approved CAR-19 therapies for B cell malignancies. And so we considered maybe this could be a secondary indication, but more importantly, this strategy would target both peripheral B cells as well as those that are in your bone marrow, your spleen, potentially lymphoid organs, etc. So we used this approach briefly. We took wild type mice, we treated them with AAV vector so that they would develop AAV neutralizing antibodies. And then we treated them with CAR-19 and then collected their blood to see if this was effective at getting rid of AAV neutralizing antibodies. So the first point I just wanted to point out is that when we took animals and we treated them with the CAR-19 vector, this is quantifying peripheral CD19 positive B cells. You can see the CAR-19 mice did no longer had the CD19 positive B cells in the peripheral compartment. And then, of course, the animals that didn't get CAR-19 did have those B cells. They go down here, and this corresponds with the timing that they get radiation so that you can give the CAR T cells. But importantly, what the CAR-19 was effective at getting rid of CD19 positive peripheral B cells, but also those same CD19 positive B cells in the marrow and spleen. And then when we quantified, so these animals have been pre-immunized with AAV and had very high titer neutralizing antibodies to the infused AAV serotype. But when the animals were treated with the CAR-19, those AAV neutralizing antibodies decrease on average to a titer of roughly 1 to 5. And then, of course, as you might expect, NTD stands for non-transduced, basically not treated with CAR-19. The titers did not decline and actually continue to rise. So in summary, these CD19-based CAR, we think may reduce or eradicate high titer neutralizing antibodies to potentially permit efficacy after systemic AAV vector administration. And actually to just test this point, at least in this model, these are the data I just showed you, but briefly, we wanted to basically see after you treat these animals, you pre-immunize them with AAV, so they have AAV neutralizing antibodies, you treat them with the CAR-19 vector. We know that gets rid of the AAV neutralizing antibody or markedly reduces it, but then does this permit efficacy of AAV vector administration? And that's what these data are. So we treat these mice that had received CAR-19 therapy versus not with an AAV vector, a second dose of AAV, and looked at expression of factor VIII. So these are animals that had AAV neutralizing antibodies, but didn't receive the CAR-19 therapy. So went through the same controls, but didn't have CAR-19. These are the animals that were not pre-immunized. So at baseline, they had no AAV neutralizing antibodies, but did go through the same process of irradiation and CAR-19 therapy. This is their level of expression. And importantly, the animals that had pre-immunized, so initially had AAV neutralizing antibodies that went down, their factor VIII expression actually was the same. So this suggested that this approach, at least in this model, effectively permitted repeat AAV vector administration. So then we wanted to compare CAR-19, which really can target peripheral B cells as well as lymphoid compartments and lymphoid organs that harbor B cells, relative to monoclonal antibody-based approaches that one may anticipate largely target peripheral B cells. So we did this with a couple different monoclonal antibody-based approaches. So we used this anti-B2220. This targets CD19 as well as CD20. So both these compartments of B cells. And then CD20, which is akin to rituximab, targeting this B cell compartment. So briefly, as you may expect, this is flow data. As it may expect, the CD20 antibody got rid of peripheral CD20 positive B cells, and then the combined antibody reduced but didn't eliminate these cells. But importantly, the monoclonal antibody-based approach really had no effect on AAV neutralizing antibodies. So these animals were pre-immunized with AAV, and then this is their AAV neutralizing antibody titer after treatment with the antibody-based approach. And I think what is important here is that I think this shows us the compartments of B cells that we may need to target. So this is the peripheral B cell compartments that were reduced with this monoclonal antibody-based approach. But notably, these animals, when we looked at their marrow and spleen, the B cell compartments were essentially unchanged relative to treatment with an isotype control. So what we think, as I've mentioned a couple of times, is that in order to get rid of these AAV neutralizing antibodies, one approach may be that you have to target both peripheral B cells as well as B cells in the marrow and lymphoid organs and in the spleen. And this may effectively get rid of neutralizing antibodies or markedly reduce them to permit repeat AAV vector administration. We were not able to achieve this with use of monoclonal antibody-based approaches. And then I'll just say any translational efforts, I think it may be a tall order to treat somebody with a CARB-19 based approach and then subsequently an AAV vector. However, there's quite a lot of innovation in the CARB approaches that may permit either transient B cell depletion as well as potentially using approaches that do not require myeloablative conditioning and therefore would be much safer. So anyways, we hope that this contributes to the body of work that may think about future efforts on how to get rid of neutralizing antibodies. And then lastly, I just want to close with just thinking about where we may be going in the future and where the use of adeno-associated viral vectors may or may not be important. So I wanted to know one point. Hemophilia A is caused by thousands of mutations. This is just looking at mutations per 100 base pairs in the different domains of factor VIII. And at this point, from a regulatory standpoint, for better or for worse, you cannot really develop a therapeutic for every one of these individual mutations. And so I think first generation efforts, for example, for gene editing, are going to require a mutation agnostic approach. And some of that concept is being actually pursued actively now for hemophilia B with open clinical trials. So the idea is for hemophilia B is you integrate the entire factor IX cDNA into a safe harbor locus. And a similar approach for factor VIII, I think, may be the next initial approach to hemophilia A gene editing, although hard to predict the future. But I think what's really important when thinking about future approaches for either gene addition as well as initial efforts at gene editing, which I've sort of noted are likely to require knock-in of the entire factor VIII gene, there's really only one effective way to deliver a large piece of DNA to the nucleus, and that's AAV vectors. And so I think there's a number of different non-viral based approaches that are in development. This is lipid nanoparticles or viral-like particles, and these are obviously very exciting. But they hold the challenge that they don't efficiently and safely deliver DNA to the nucleus, whereas AAV we know can. And so I think there's some reasonably founded sentiment that the path forward to overcome current gene addition or future gene editing limitations may be to sidestep the use of AAV vectors. But I think the reality is that I think safely delivering DNA to the nucleus, I anticipate we'll be using AAV vectors for the foreseeable future. So then I think that begs the question of, in current progress, when you start to, I think the current progress is really outlined in the next series of questions that we need to overcome to improve on next generation efforts, which are outlined here. Some of them are outlined here anyways. And then the question is, are these challenges likely to be specific to AAV? So I've given this my best assessment. Obviously, I don't have a crystal ball to foresee the future, but when you look at questions around durability of expression, this is certainly an outstanding question for hemophilia A, but not as much for hemophilia B. I think ultimately, if it's related to the amount of factor VIII protein you're expressing, this is unlikely to be AAV specific or even gene addition specific. The variability in expression, I sort of noted briefly the number of biological variables that are, from the time of systemic infusion to maintaining a stable piece of DNA in the nucleus for factor VIII or IX expression. But that is, I think that there's going to be a number of possible places for biological variability, likely independent of the method of how you deliver this. So I think some degree of variability expression is ultimately going to have to be tolerated by all platform-based approaches for gene therapy. We've seen some unpredictability of the individual response. I don't necessarily think that's AAV specific. This concept, do all patients have therapeutic benefit? This is, I think, difficult to answer if this is AAV related or not, and one may anticipate you'll see this in other approaches. We don't have time to talk about long-term safety, but clearly we don't yet know the long-term safety of AAV, although there's a mounting body of evidence. AAV's been in clinical development for gene therapy for over 25 years. But this point around AAV neutralizing antibodies, I do think is a sticking point that may end up not being seen in approaches outside of AAV. So this inability to redose is certainly a limitation right now for AAV, and this may end up being specific to AAV and, for example, not observed with lipid nanoparticles or other approaches. So I'll just summarize. I hope this wasn't too much of a whirlwind, but I just summarized what we reviewed. So we talked about factor VIII expression durability. Just to highlight again that factor VIII expression in the range of moderate or mild hemophilia has been observed in animals and in people. We think the mouse model may be a useful model to further study the mechanism of factor VIII expression durability. Thus far, the data support the initial loss of high factor VIII expression is due at least in part to the loss of vector copy number. When you think about available clinical data as well as the data I present around mechanism of factor VIII durability, I think this supports the potential long-term efficacy of incorporating enhanced function factor VIII variant with the concept again that you could approximately normalize factor VIII expression with low levels of expression that may be important for maintaining long-term expression and durability of expression. And actually, this approach is even this week we learned is being pursued by Spark Therapeutics in a phase 2b trial. I think the universal development high titer neutralizing antibodies after systemic AAV expression and the likely inability to redose, or there's no proven strategy to redose, I think highlights the importance of expression durability. And then we talked about some potential approaches of targeting B cells and the various B cell compartments that may need to be targeted to achieve meaningful reduction of AAV neutralizing antibodies. And then lastly, I think an important contextual point of where we are in the field is that AAV is really, at this point, the only efficient way to safely deliver DNA in vivo to the nucleus. And so I think, in my opinion, I think AAV is likely to be critical to the success of hemophilia gene therapy for the foreseeable future. I want to thank my lab, who's outlined here, particularly those that I've highlighted have been important for some of the data I outlined here. And then lastly, I just wanted to note that myself and others have developed a novel clinic specifically for patients that may be interested in hemophilia gene therapy and or have undiagnosed coagulation disorders. We do have experience with commercial as well as decades of experience in research hemophilia gene therapy, and we certainly would be happy to talk to anybody that may be interested in some education or have questions around this. So with that, I thank you for your time, and I'm happy to take questions.
Thank you very much, Dr. George. That's a really interesting update on something that I think we've been following for a couple of years now, and really exciting to see, where we are now and think about where this might be going next. So thank you very much for that. We'll dive right into our Q&A with Dr. George now. So if you have any questions for her, please enter them in the Q&A feature at the bottom of your screen, and we'll do our best to answer all of your questions. Maybe I can start with kind of a big question, kind of a standing back and looking at this from a little bit of a distance question. Can you maybe talk about how this research and these advances and this current, analysis of where we are might play into the sort of shared decision-making conversations that people with hemophilia A are having with their healthcare providers, and thinking also about, this is some pretty granular research. Are there any tools to help lived experience experts, people with hemophilia, to understand this such that they can be really well equipped to have those conversations?
Yeah, no, it's a great point. I mean, in some ways, that there's so much activity in the space. I mean, it's obviously very exciting, and I think makes me very optimistic that we'll continue to only get better at this. But it can also be a lot of information to take in and to keep track of, so to speak. I think I'll just start with the first part of your question. I mean, I think that part of the shared decision making and really the importance of, I mean, I would, the decision to pursue gene therapy, I think, and when I've spoken to patients about this, it involves multiple conversations. And I think the critical thing is making sure you know what you know, what you know, don't know, what you know, and what you don't know, so to speak. And there are multiple things we don't know yet, and just sort of delineate them, and then the supporting data to help us generate where this, even though we don't have direct answers, we do have supporting information to point us in one direction or another. But I think making that decision, the critical pieces I would encourage people to pay attention to is trying to particularly if it's a licensed vector, having a real conversation about what the outcomes were of the clinical trials, acknowledging this role of AAV, multi-year persistent AAV neutralizing antibodies is like to preclude repeat administration. But understanding the clinical trial data about durability of expression, what was the variability, what were some of the outcomes, what were the safety measures, what was the use of immune modulation? Because I think it's there are certain points about this work that are generalizable, but there's quite a lot of nuances with each of the individual vectors. And so I think you have to build the foundation understanding what AAV gene therapy is, but then I think you have to build upon that with the individual components of what we know about the vector that patients may choose to pursue, either from a licensed therapy or even on the clinical trials. Data becomes available throughout the trial that I think is important in the informed consent that people are aware of.
Right. Yeah. There's a lot of different things to try and follow. A lot of really complex things for people who maybe are not steeped in this environment to try and wrap their minds around. One interesting question that we actually had submitted in advance by somebody was to ask whether gene therapy requires lifelong immunosuppression.
Yeah, it's a good question. I mean, to my knowledge, no, there's not any, not in the hemophilia space certainly, that have required lifelong, immunosuppression thus far. Of course, there is quite a bit of variability in the trials in terms of the relative use of, prevalence of patients that required immune modulation, as well as the duration. I think for some time it was being described as a short course of immune modulation. The reality is that most of the patients that are put on immune modulatory strategy is typically not a short course. It's multiple weeks to multiple months. But there's so much variability in terms of what's used, the number of the percentage of patients that require it, and then the duration of use, which I think goes back to this point of it's best to just understand the specific vector that you're maybe receiving to understand that.
And maybe a related question then is I know that the different vectors can be related to different AAV serotypes. So does the immunity, is there sort of cross immunity? If you have immunity, let's say to AAV2, are you maybe still a candidate for an AAV5 based vector, or does it kind of go across the serotypes?
Yeah, so thus far, the available data suggests that once you receive an AAV vector, you develop AAV neutralizing antibodies certain to the serotype that you receive, but it appears to be multi-serotype cross-reactive. So you're absolutely right. Like, would a strategy be to sort of engineer an AAV capsid that just the antibodies couldn't recognize, for example? Certainly there's people that have worked on this previously and continue to do so, but that has not, there's no evidence of that thus far. So I guess what I would say is I would make the data thus far conclude that if you receive a systemic AAV vector, it's likely to be cross-reactive, at least with all known serotypes.
Right.
But I want to emphasize, we haven't tested this hypothesis. Like, there's, to my knowledge, now somebody has received a second AAV vector, but I think it's reasonably supported that it would not be efficacious.
And does that also count, or does that also hold true if you have a pre-existing immunity? Because I think you can have been exposed to these viruses already, and even if you've not had gene therapy.
Yes, good point. So, there are buccal data of environmental, AAV is naturally occurring in the environment. The natural serotype you're exposed to is AAV2. The trials in hemophilia are not using AAV2, but most people that have environmental exposure, the serotype, just as when you receive an AAV vector, when you have environmental exposure, typically they're cross-react with multiple serotypes. So The exact titer may vary, but in most cases, if you have a neutralizing antibody to one serotype, you're likely to have it to another one.
And maybe I'll take this conversation a little bit of a different direction. We just had a question from an audience member, so I'll just shelve my question for a moment because I just liked asking my questions, but so this is coming from Dr. Glenn Pierce, and he commends you on an excellent talk synthesizing a lot of data. And he wanted to mention that the WFH shared decision-making tool to help people consider the treatment options available is at sdm.wfh.org. And the WFH Gene Therapy Registry exists, and this is intending to monitor the unknowns long-term after gene therapy. So those are really good resources for our audience to be aware of. That tool again is at sdm.wfh.org. And the Gene Therapy Registry is something that's international and looking to register if possible. I think they're hoping to include everybody who gets gene therapy in the world, right? That's the goal of that registry.
Yeah, and I'd say actually, if I may just comment, like expand on that a little bit. Glenn, thank you very much for sharing that. The registry, so one of the roles I have is we organize gene therapy efforts across CHOP, and the hemophilia community is particularly organized, and much to Glenn's leadership in that piece. So this registry, I think I'm quite excited about because and optimistic that there'll be some information that'll be really important for hemophilia patients, the hemophilia community. Because to my knowledge, I'm unaware of other advocacy organizations that have collectively gathered their patients in one place that may be receiving AAV vectors from multiple different sponsors and manufacturers. So what we would be worried about around, for example, long-term safety are theoretical, what are expected to be rare or ultra-rare safety events. So you really need to have collated data from a large cohort like we hope to achieve with the WFH registry.
Right. Yeah, because the clinical trials don't generally have hundreds of people in them, do they, at the moment?
No. And the other, I think, important point for people to recognize is right now for the clinical trials, the required FDA in the United States anyways, required follow-up is 5 years. Most of the licensed vectors now have committed to post-marketing surveillance studies for up to 15 years. So it's not lifelong required monitoring. And so I think that registry would be really important for understanding long-term safety.
Great, thank you. Oh, and I was waiting for this. We've got a question from Lawrence Woolard in the UK. I saw he was on there. I knew he had a question. He also wanted to thank you for the talk. Let's see what he's saying. So the commercial launch of gene therapy for factor VIII has come under increased scrutiny due to minimal or slow uptake. Does Dr. George have any observations here beyond general market access challenges? And what does she think this means for the development of next generation AAV gene therapy for factor VIII, given the commercial pressures? And I'm sorry not to give you a lot of time to answer but I think that is a really important point to touch on, that there's clearly interest from the community, but there seems to be some issue here with getting that value across, I guess, to payers. Is that maybe where we're
Well, I mean, I haven't, my assessment so I don't know in Europe, but in the United States, I think some of hasn't necessarily been an access, but rather interest on the patients, because it's hard to be the first, right? But people have a lot of questions on the clinical trial data that have come out. And then they also know there's a lot of progress in this space. And so there hasn't, I think it might be a combination of access as well as enthusiasm. Which I think is different than observations for hemophilia B, which has really, in my observation so far, been more of an access question. So I know I only have 30 seconds, but that's my answer.
Okay, well, thank you. I appreciate that. I think the context in the UK, and I don't want to say anything too definitive, but I think there have been some applications for coverage by the healthcare system and some difficulties there with getting those approved. So I think that might be what Lawrence is referring to, but I do apologize if that's not the case. And of course, the payment systems are so different in different countries that then, of course, the context can vary so much. But I'm afraid that we are out of time, but I do want to thank you very much, Dr. George, for this really interesting presentation and this discussion. We could have clearly continued discussing for quite a while, yet I still have a list of questions myself. So we'll have to have you back in 2025 to tell us some more. So thank you very much, Dr. George. I'd also like to thank our audience for being with us today. The webinar will be available on the NBDF website, bleeding.org, shortly after we finish here. Under the events tab, there you can find all of our archived webinars, and also keep an eye on that events tab for the schedule of upcoming Wednesday webinars in January. So thank you so much for joining us for this last Wednesday webinar of 2024. I wish everybody a restful, enjoyable break and look forward to connecting again in 2025.